Hi, my name is Sarah Davis, and I'm a graduate student in the Project Dragonfly Advanced Inquiry Program. My master's research has been focused on developing and managing a citizen science initiative called Mapping Microplastics. The goal of the project is to engage a wide range of audiences in testing their local waterways for evidence of microplastic pollution using rapid staining and fluorescence. Plastic pollution has become a critical focus for conservation scientists. Microplastics are defined as plastic fragments smaller than 5 millimeters in length and have been identified in nearly every habitat on the planet. The majority of microplastic particles will never biodegrade and present a huge problem for our Earth's oceans, which are the endpoint for most waste. In aquatic ecosystems, microplastic pollution has been documented to create significant ecological problems. While the presence and abundance of microplastics has been studied in marine ecosystems, it remains largely under-researched in most waterways. This project collects critical baseline data about where microplastics are and how dense the pollution problem is in different areas. From the preliminary data that's been collected, microplastics have been most dense in urban adjacent waterways, but present in all bodies of water surveyed. So why citizen science? There are two major reasons, one being efficiency in data collection. Citizen science researchers can gather a huge amount of valuable data across a large geographic area that would cost more formal research methods much more time and funding to collect. The second reason is that at its core, I developed mapping microplastics as an educational tool. For many, the science research community can feel like a place where only experts are welcome. As a woman in science, I'm passionate about creating opportunities for historically underrepresented groups to contribute to real important research. Experiences like this are also demonstrated to help shape kids' identities as scientists and encourage participation in STEM well past the activity's completion. I developed a simple procedure from published lab studies and with feedback from microplastics and fluorescence experts. While studies recovering microplastics in environmental samples are relatively new, there's really no formal agreed upon best practice, but the literature supports Nile red staining as a way to quickly and effectively test for larger microplastic pieces, generally between 1 and 5 millimeters in size. While identification techniques like FTIR and Raman spectroscopy can offer a more sophisticated and precise identification of recovered plastics, there's sufficient evidence to suggest that Nile red can be a useful tool for taking a quick and dirty assessment, so to speak. So how does it work? First, participants simply scoop up a water sample from a local waterway. Next, they remove all the solid debris using a coffee filter. The next step is to apply Nile red stain, which adheres to plastic fragments and creates fluorescence. After allowing the stain to permeate, participants view the sample under blue light with an orange filter and record any fluorescing microplastics that are present. They're also required to take a photo in order for the results to be validated. The last step is sharing the results. Participants upload their data to the project's website, where it's reviewed and added to an interactive GIS map. There are two major methods of evaluation that I think are critical for the project's success. The first is a participant impact study, which is currently recruiting participants with an anticipated completion date of December and data analysis occurring in the spring of next year. Participants will complete a pre and post activity survey to evaluate their experience with the citizen science project. This survey will evaluate three major themes: connection to local natural areas, understanding of the plastics crisis, and connection to science research. The second evaluation component is a study to validate the citizen science methods. The nature of the program I'm part of is that it's predominantly virtual, so I do not currently have access to the lab equipment in order to complete this evaluation. However, I think it could be accomplished by collecting a subsample of the filter papers produced by volunteers and verifying their results using FTIR or Raman spectroscopy. This would offer important information on the reliability and accuracy of the data being collected by citizen science volunteers. Lastly, I'd like to sincerely thank my advisors at Project Dragonfly, Miami University, and the Wildlife Conservation Society, as well as the Miami Women Giving Circle for their funding support. My email is listed here if you'd like to communicate with me about the project, as well as the Mapping Microplastics website. Thank you very much for your time.